Jacobin monad to essentially dissect all of the different components here and to figure out which elements were binding to which sites, etc. So what I would like you to do is to now think about which proteins or DNA elements could be mutated to cause an uninducible operon, and which proteins or DNA elements could be mutated to cause a constitutive operon. So think about these. You definitely will need to be able to identify some mutants that would give you uninducible operon that cannot be turned on ever, or a constitutive or constantly turned on operon. Okay, a little bit of extra content. I like to bring this up because we will be going into the biotechnology chapters in the next couple of weeks. And lac operon, in addition to providing us with sort of the general model for how gene regulation can work in bacteria, has also provided us with a very useful tool in biotechnology. And so you often will see applications, lab applications that use this lac operon and its components. So let's take a little bit of a closer look at how this can be used in the lab. So one place where many scientists have sort of co-opted the lac operon is in the synthesis of proteins. So if someone, for example, wants to produce, let's just say they want to produce insulin. They're interested in using bacteria to produce insulin that can then be purified and injected into humans who have type 1 diabetes. So to do this, what you can do is to take your gene of interest, let's just say take insulin, gene X, and you can clone it downstream of the lac regulatory region. So that is basically where the operator, promoter, etc. site are located. You can then add an inducer, and rather than adding lactose to the cell, you can actually add another type of inducer, which is an analog. It also induces, but it is not metabolized by beta-galactosidase, and that is this reagent called IPTG. We'll look a little bit at how this works in a couple of slides. So the way that you would set the system up is, let's say you clone your gene. You could say insulin, or here they give the example of human growth hormone. You put that gene into a plasmid vector. You have the promoter from the lac control region, and you then clone this and put this plasmid into a bacterial cell. So you use a process called transformation. You put that plasmid inside, and this, of course, is the genomic DNA of the E. coli cell. If I then want to induce this protein, or this protein to be made, I will add the inducer. In this case, I could add either lactose or I could add IPTG. And in the case of adding this, I get transcription of this gene, I get the messenger RNA, and I get my protein made. So pretty soon I have a whole bunch of bacteria with a whole bunch of protein, and I can lyse those bacteria open and purify out the protein that I want. So this is incredibly useful for quick induction of protein. And the reason you would use this system is that you don't want the gene to start to be made before you've grown these cells up to a particular density. So this allows you to add IPTG once you have your bacteria growing well at a particular density. Okay, so let's look at a couple other components here. I mentioned that IPTG can be used as an inducer. It's considered an artificial inducer. This is the structure of IPTG. And what you can see is that it somewhat resembles lactose. So lactose would have a galactose and a glucose molecule. This acts in exactly the same way. So it acts exactly the same as lactose does, except for the fact that the IPTG is not a substrate of beta-galactosidase. So it will induce by binding to the repressor, getting the repressor to come off of the operator, but it is not processed by beta-galactosidase. Okay, so this chart right here shows you an induction, the time after you've added IPTG, and this measures the lac operon messenger RNA that has been produced. So you can see that you can get very robust induction of the lac operon through the addition of IPTG. So many people will use this because it's easy to add this artificial inducer in place of lactose. The second way that people often use the lac operon is, instead of using the gene regulatory elements from the lac operon itself, they will often use one of the genes, the LAC-Z gene, which, as you might recall, produces beta-galactosidase. So the way that this is often used or applied is that if people are interested in studying, let's say, where their gene of interest, whatever gene X is turned on. So they want to basically look for a transcriptional reporter. Where is this gene turned on? They can clone LAC-Z right downstream of gene X regulatory region. Okay. And so what will then happen is that wherever their gene is expressed, whatever cells, whatever tissue types, those cells will produce, instead of the actual gene X, will produce this LAC-Z protein product, beta-galactosidase. And there are ways to then measure beta-galactosidase activity. There's a particular substrate that you can give to beta-galactosidase that produces a blue color, and that is called XGAL. So this XGAL, if you put this into the medium, if beta-galactosidase is present, it processes this XGAL into a colored blue product. So whenever you see blue, you know that beta-galactosidase has been expressed. I'm going to come back to this slide in just a moment, but to walk through this particular experiment was an experiment where they were trying to look at which cell types and tissue types a particular promoter was turned on for this PROM1 gene. This was a stem cell, I believe a stem cell marker. So this PROM1 gene is a stem cell gene that gets turned on in stem cells. And so what they did is this: they fused the PROM1 promoter to a LAC-Z reporter, and then they treated this with this XGAL substrate so that they could see which parts of this mouse had the expression of the PROM1 gene. And they were able to identify which cell types in either the neonatal or the adult were expressing this stem cell gene. Okay, so that's one way that you can use this. Another way that we used to do this a lot more, but XGAL used to be used in cloning. And essentially the way this works is that you would have a plasmid, which would be, imagine that this is the plasmid, and there would be a cloning site in the middle of the LAC-Z gene. So if you were successful in cloning your gene into it, you would disrupt the LAC-Z gene, and you would not produce any functional enzyme, and therefore you would get a white colony. If you are trying to clone something, you want to get that gene cloned in there. So this is the result that you would want to see. And so on your plate, you would get a whole bunch of colonies. Some would be blue, some would be white. And so if you want to get the colonies that have a bacteria with a plasmid in which you have been able to clone your gene, which color would you want to pick? You'd want to pick the white colonies. and those ones would successfully have cloned your gene into that cloning site. If you ever saw a blue colony, you would know that nothing had been inserted into the lac-C gene, so you got the functional enzyme, which can then process XGAL into this blue color. Okay, so that completes our gene regulation in prokaryotes lecture.